Hello judges, my name is Julia Seymour.
And I'm Rylan Donohoe.
We are both senior students from St. Ignatius of Loyola Catholic Secondary School in Oakville, Ontario. Before we share our project, we would like to sincerely thank you in advance for listening to our presentation. We hope that you and your families are staying safe during these times.
From shifting weather patterns to rising sea levels, climate change is the defining issue of modern society. Without immediate intervention, scientists have predicted that the impacts of climate change will become irreversible within 12 years. Completely transitioning to renewable sources of energy, whether that be solar, wind-powered, or hydroelectric, lies at the core of environmental sustainability efforts.
Investing in biofuels is a rapidly emerging solution to this environmental crisis. Corn and sugarcane have conventionally been used as the primary sources for bioethanol production. However, their long-term use, both as a food and fuel source, is unsustainable. Researchers are, therefore, currently investigating alternative methods for bioethanol production, namely lignocellulosic biomass, or LCB, which includes grasses, leaves, and the non-edible parts of crops. Not only is LCB by far the most abundant bioresource on the planet, but being a second-generation feedstock, that is, a non-food material, gives LCB an immediate advantage over conventional bioethanol resources.
Despite its immense potential, the industrial application of LCB as a renewable source of energy is thwarted by its recalcitrant molecular structure. As seen in Figure 1, the cell wall of LCB is composed of three rigid polymers tightly bonded to one another by hydrogen and van der Waals bonds. They include 40 to 50% cellulose, 25 to 35% hemicellulose, and 15 to 20% lignin. Cellulose is the primary source of fermentable sugars in LCB and must therefore be easily accessible to facilitate its conversion to bioethanol. However, the strong entanglement of lignin molecules amplifies the recalcitrant structure of LCB, which dramatically decreases the efficacy of subsequent enzymatic hydrolysis and ethanol fermentation.
Researchers are continuously developing methods that can break down the rigid molecular bonds of LCB. Scientists have proposed acid and alkali treatments, among others. However, all of these methods require large quantities of chemicals and/or long processing durations. A method that is both economically and environmentally sustainable has yet to be established. Our project aimed to develop a novel lignocellulosic pretreatment method that could overcome these barriers and achieve the most efficient fractionation of LCB, thereby maximizing the bioethanol conversion rate for these non-traditional sources of energy.
Many research teams are currently investigating the effects of microwave irradiation on the delignification of LCB, while others are focusing on ultrasonication. While microwave irradiation induces quick and controlled heating for an optimal cellulose recovery of 93.05%, ultrasonication relies on the principle of cavitation to reduce hydrolysis time by up to 80%. Upon further analysis of both these technologies, the drawbacks of each appear to be advantages of the other.
Rylan and I, therefore, wondered if a possible synergistic relationship existed between these two pretreatment methods. As such, the purpose of our project was to determine the microwave power in watts and duration of submerged ultrasonication in seconds that, when applied together, would result in the greatest bioethanol conversion rate from corrugated fiberboard. In turn, this would alleviate current economic and environmental burdens of pretreatment processes, thereby optimizing LCB's potential as a source of renewable energy.
Based on past experiments, we hypothesized that a microwave power of 1100 watts and a duration of submerged ultrasonication of 480 seconds would result in the greatest bioethanol conversion rate from corrugated fiberboard. This would allow for optimal fractionation of LCB with minimal processing times and inhibitor formation.
To test our hypothesis, the experimental procedure was divided into 3 phases.
During the first phase, the samples were milled, microwaved at power settings ranging from 0 to 1100 watts, and exposed to ultrasounds in a submerged medium for a duration ranging from 0 to 480 seconds. A cellulase enzymatic treatment was subsequently added to the samples to effectuate the enzymatic hydrolysis of the exposed cellulosic filaments into their glucose monomers.
After a 24-hour incubation period at 40 degrees Celsius, the second phase of the experiment began and consisted of adding 5.3 grams of Saccharomyces cerevisiae, commonly known as household yeast, to each of the samples. After being thoroughly mixed with a magnetic stirrer, the samples were secured with a water-sealed airlock and placed back into the incubator for another 24 hours to facilitate the process of ethanol fermentation.
Once the second incubation period was complete, the bioethanol concentration of each of the samples were quantified using a passport ethanol sensor calibrated to a self-diluted 1% volume-volume solution of ethanol.
The experimental procedure outlined above was completed on 3 different occasions. Between each of these trials, key procedural modifications were made, such as increasing the duration of the milling process and decreasing the initial dimensions of the lignocellulosic square prisms. These modifications were made in an effort to achieve maximal delignification of the corrugated fiberboard before commencing formal experimentation of the independent variables.
In the end, the combination of selected values for microwave power and duration of submerged ultrasonication resulted in an array of bioethanol conversion rates, some of which disproved our initial hypothesis. Before examining the possible synergistic relationship between microwave and ultrasonic irradiation, the independent variables were analyzed individually.
Using computerized curve fitting on the MATLAB programming software, a polynomial equation to the second degree best represents a correlation between microwave power and ethanol concentration. The vertex of the graph indicates that a power setting of 555 watts results in the greatest theoretical bioethanol conversion rate at a concentration of 1.91%.
The correlation between duration of submerged ultrasonication and ethanol concentration is best represented by a rational equation with polynomials to the first degree in both the numerator and denominator. The horizontal asymptote of this graph, situated at y equals 1.95, indicates that ethanol concentration continually increases to 1.95% as duration of submerged ultrasonication approaches positive infinity.
Afterwards, we analyzed the relationship between microwave and ultrasonic irradiation using a three-dimensional Cartesian coordinate system. All of the experimental data points were plotted in the first octant and fitted with a thin plate spline curve for data interpolation and smoothing. A thorough analysis of the data collected revealed a theoretical peak of 2.26% at a microwave power of 500 watts and an ultrasonic duration of 462 seconds. For better visualization of the patterns in the XY plane, a corresponding contour plot was created. This two-dimensional graph displays an unambiguous peak that confirms the synergistic relationship that exists between microwaves and ultrasounds regarding ethanol production.
This novel lignocellulosic pretreatment method resulted in a bioethanol conversion rate that is 0.06% greater than that of untreated starch derivatives. This is a significant discovery since ethanol yields from untreated starch derivatives have historically surpassed those of lignocellulosic sources by about 0.50%. While this may seem statistically insignificant, 0.06% represents billions of gallons of bioethanol when applied on an industrial scale. Moreover, with the price of starch-based biofuels approaching an all-time high as seen on this graph, the application of this new pretreatment method truly has the potential to revolutionize conventional practices in the bioethanol production industry.
By researching strategies to further optimize our novel lignocellulosic pretreatment method, we firmly believe that this new approach for industrial bioethanol production can and will lead to a more sustainable tomorrow.
To conclude, we would like to take this opportunity to offer our utmost gratitude to our teacher supervisor and mentor, Mrs. Erin Stewart, for her unwavering support throughout our project. We would also like to thank and recognize Mr. McCarthy and the Department of Science at St. Ignatius of Loyola for providing us with most of the materials needed to perform our experiment.
Thank you for listening to our presentation and have a wonderful day.